Okay, everybody, let's talk about uveal melanoma clinical trials. I'm going to first go over some completed trials and then jump into some ongoing studies. One trial that completed enrollment is the Adjuvant Crizotinib study. This was a study for high risk or class 2 patients to take in their first year after being diagnosed and treated the oral therapy Crizotinib. And the study was reported this year at ASCO 2020. We saw that the patients who took Crizotinib did not have an improvement in relapse-free survival or overall survival. So we call this essentially a negative study. So we're still looking for the proper adjuvant treatment for high-risk uveal melanoma patients. We finished and published a study using ipilimumab and nivolumab for metastatic uveal melanoma. And this was published in the Journal of Clinical Oncology at the end of October 2020. And what we saw in this study is that nivolumab plus ipilimumab combination immune checkpoint demonstrates an overall response rate of 18% and a duration of response of 12.1 months, with an overall survival of 19.1 months in this population. Majority of the patients that we treated on this study were treatment naive. They had not seen other therapy for metastatic disease, but 2 patients had seen prior therapy, and one of those patients had prolonged stable disease or clinical benefit from the treatment. Moving on, another study that completed enrollment is the FOCUS study. FOCUS is the revised single-arm trial of percutaneous hepatic perfusion with melphalan. And patients undergo up to 6 treatments and then are followed for response and survival. Here's a slide of responses in uveal melanoma, where you see hypervascular tumors noted by the arrowheads on the left, and what is seen as a response to treatment on the right. Another study that has completed enrollment involves the use of ImmTACs. ImmTACs are immune mobilizing monoclonal T-cell receptors against cancer. And ImmTACs can be made against a number of tumor proteins. They are bispecific in that one end recognizes the tumor molecule, the other end recognizes T cells, essentially magnetizes T cells over to the tumor environment. You see in the first block in panel 1, you see a tumor protein being presented in green in the context of an HLA molecule. The HLA is the green molecule, the A0201 that some of you might be familiar with. And then the orange is actually a piece of the cancer protein that's being presented. The drug is the blue. And so what you see in panel 2 is the drug binds to that protein that's presented in the context of the HLA. And then what you see in panel 3, the back end of that molecule is attracting T cells. It's essentially got a magnet or a receptor, or a binder, I should say, for the T cell receptor. So it kind of lures the T cells over, locks in with the orange T cell receptor, and that stimulates activation of the T cells, which leads then to tumor cell killing, release of enzymes, granzymes, etc. That's panel 4. So the most well-studied ImmTAC to date in uveal melanoma is the drug tebentafusp. Many people know it as the Immunocore drug. This molecule is HLA A0201 restricted, and it's a GP1 targeting drug. And so what you see on the left there is, again, if you think of the cancer cell on the top, the HLA molecules are the green molecules, so that would be A0201, presenting a piece of tumor protein, that red sort of curved little speck. That little speck of tumor protein is being presented in the pocket of HLA A0201 just so, and that binds the ImmTAC molecule nicely. That binding essentially then gets the ImmTAC molecule to use that corkscrew end that it has to recruit over T cells. And that corkscrew end of the ImmTAC molecule then fits in the pocket of a T cell receptor. And in linking there starts a T cell activation cascade. So this study that completed this year is a randomized phase 2 study, and it compared tebentafusp against investigators' choice therapy, essentially chemotherapy with dacarbazine, ipilimumab, or pembrolizumab. And this study was for treatment-naive uveal melanoma patients. And we anxiously await results from this one. Let's move on to ongoing studies. The targeted therapy protein kinase C inhibitor IDE196, it's currently being studied in combination with a MEK inhibitor known as binimetinib. Protein kinase C is involved in cell signaling. It starts a cascade just under the GNAQ, GNA11 cytoplasmic tail end and leads to downstream signaling and, of course, downstream of protein kinase C, which I'm moving my mouse around here, circling. Downstream of that is the MEK protein. So the study is targeting both MEK and PKC, ongoing and open to enrollment. Another study is using focal adhesion kinase inhibitors. Focal adhesion kinases play a role in the tight junctions of the glomerulus of the kidney, so they work in your kidney cells, and they regulate something called a YAP signaling and the Hippo pathway. And these are active in uveal melanoma. You see over on the far left, again, this is your GNAQ, GNA11 purple molecule with 7 transmembrane arms, if you will, or domains. And that essentially, the GNAQ, GNA11 kicks off a row cascade, which eventually signals down to YAP and what's called the Hippo pathway. There's a protein called FAC in there that helps with this signaling. And so FAC inhibition essentially would decrease the ability of a YAP to move itself over to the nucleus. So a reasonable target in uveal melanoma. The study is open and enrolling. And in fact, the company is combining their FAC inhibitor with the MEK inhibitor cobimetinib. And the study is enrolling worldwide. There is a nuclear transport protein inhibitor known as selinexor. This drug is FDA approved for multiple myeloma, and it has a rational strategy in melanoma as well. And so there is a cohort for uveal melanoma where patients receive selinexor, an oral pill twice a week in combination with a PD-1 inhibitor in uveal melanoma. These nuclear exportins, if you will, this XPO, these nuclear exportins remove tumor suppressors from the nucleus. Tumor suppressors are actually a good thing. You want them around to suppress tumor. And so exportins are actually taking them out of the nucleus. And what selinexor does is it blocks that exportation. So it keeps the tumor suppressors back in the nucleus where they can essentially do their job, restore their function. So this would theoretically lead to tumor cell death. In tumor cell death, we think then that makes for rational presentation to the immune system, and so hence the combination with PD-1 therapy. Another ImmTAC that's under development is one against PRAME. And so for PRAME-positive tumors, not just uveal melanoma, but some other PRAME-positive tumors, there's another ImmTAC being developed by Immunocore. And this ImmTAC is targeting, again, PRAME. This will be interesting to see if this functions the way it's working right now against GP100, in that it seems to attract the T cells over and then lead to T cell activation. By virtue of ImmTACs, how they were designed and what I explained earlier, these will always be HLA restricted. It requires that the one end of the ImmTAC molecule specifically recognize an HLA presented protein. It can't just recognize the protein in general. It can only recognize a very small sequence of the protein presented in the context of certain HLA. Intratumoral oncolytic therapy with PV10 in combination with checkpoint blockade, another study where we have seen promising results in the injected lesions. If you look on the right-hand side, the red, blue, and green plot, it's called a waterfall plot. In this waterfall plot, what you want to see is you want to see the bars going down. That indicates a decrease in tumor size from when the patient started treatment. And so what you see is in this waterfall plot, we indeed do see responses in the injected tumors in the majority of cases. And what you see above that is a type of needle approach we use. So most needles are just a single solid end, or not solid end, but an open end, a single needle. And what you see there is actually something called a quadrifuse needle, kind of looks like a broom tip. And this allows the drug to be sort of splayed out inside the tumor and not just deposited through the tip of a straight needle or a single bevel needle. So that broom-like end allows for a little bit more even or better disbursement of the drug. And what you see on the far right is PV10 is radio opaque, it's white on a CT scan. And so you see that there's nice drug retention after injection. This study is enrolling checkpoint inhibitor refractory patients. I don't know if I mentioned that. And you can essentially continue your checkpoint therapy while receiving PV10. So that's beneficial. And of course, other liver-directed therapies, electroporation. This is where you put a probe in and you signal down that probe with electrical signal. And this has the capacity to essentially rupture tumor cell membrane and kill the tumor. And on the right, what you see on the right is radio embolization, where this is one of my patients from just pre-pandemic. We treated this big, you see that dark gray area in the middle? That's a big liver tumor, metastasis from uveal melanoma. It's about 6 centimeters, pretty sizable. And we treated it with Y90, just one treatment. He didn't have multiple rounds. You really don't necessarily do multiple rounds with Y90 unless you have a lot of scattered tumor. So they deposited to this one tumor, and lo and behold, on the right, what you see in May, essentially reduction of this tumor by more than half. And in fact, I just saw this gentleman this week. It's even smaller. So he's doing really well with just one treatment to the liver, radioembolization. And a study that is planned to be open soon, according to clinicaltrials.gov, but I don't think it's recruiting. Well, it probably is recruiting. What I mean to say is I think the website is lagging behind, because I do think the study is open at the University of Miami. This is nivolumab plus relatlimab, and it's a study based on single cell RNA sequencing that was done in 3 metastatic uveal melanoma patients, where they saw a small portion of the T cell population, or the single cell population that was identified here, expressed the marker LAG3. Relatlimab is a LAG3 inhibitor. So this trial is open and enrolling. And just quickly, I want to talk about how to make T cells. Endogenous T cell therapy is pretty hot these days for a lot of reasons. And so it's a little confusing. There is a difference between TIL and endogenous T cells. So autologous T cells, your own T cells, can be harvested and delivered in a number of ways. This is essentially the idea, strawberry ice cream. How do you make strawberry ice cream? You can make strawberry flavored ice cream, right? You can have ice cream that's been flavored with strawberry, or you can have ice cream with strawberry topping, right? So both of those in some form or fashion could be considered strawberry ice cream, just kind of 2 different ways to get a very similar product. TIL, tumor infiltrating lymphocytes, is just that. So it's lymphocytes or T cells that are inside the tumor. They have infiltrated inside a tumor. So by definition, this requires surgery. It requires a tumor harvest. You have to go have a surgery or a biopsy or a superficial removal of a tumor or removal of your eye. And your T cells in there will then be harvested. And those T cells, if you think of them as these green T cells, are then enriched outside of the body, expanded, and eventually reinfused. As opposed to endogenous T cell therapy, where you have T cells against millions and millions of foreign enemies in your body. You have the potential to have 10 to the 13th T cell, different varieties of T cells. So recognizing, over, what is that? It's over 1 billion. What is 10 to the 12th? I don't know, what is 10 to the 12th? A gazillion? Trillion, billion, trillion. Anyway, over a trillion T cells in your circulating system. And in your lifetime, you're only going to call on a few 100,000 of those, but your body has the capacity to rearrange the genes and make, well over a trillion of these T cells. So that means when you're diagnosed with cancer A, B, or C, you actually have a circulating T cell that has the capacity to recognize some protein on that cancer. It's just mathematically feasible that there is, an algorithm your body has to make it. The problem is with that many options, how do you find those T cells and where are they? They're circulating, they're typically going to be in very few numbers, or they need to be enriched. And so what we do is we go fishing for the T cell of interest in the context of HLA. So endogenous T cell therapy is also HLA restricted. TIL therapy is not. We go searching for a T cell receptor that can match with a known HLA presenting a known protein in its cleft or in the context of MHC. Once those T cells are found, they are then essentially cloned outside the body, sorted and expanded again, and then very similarly reinfused. And so you see why I say it's like strawberry ice cream. You get to the same product of infusing self T cells against tumor. It's just in one situation, you don't know what proteins your T cells are recognizing, but they came from inside your tumor, so they're in the right place. They're looking around at the right enemy. In the bottom, endogenous T cell, you actually are charged with knowing what protein you're looking for, and then you go looking for T cells in the context of a known HLA and how that HLA would present that said protein, and then you enrich for those. Pros and cons to both, of course. TIL therapy, no knowledge of that tumor protein, as I mentioned, whereas endogenous cell therapy, that's a prerequisite. You must know what tumor protein you're going after because you are then going to go look for that T cell receptor that recognizes that protein in the context of HLA. TIL, no HLA restrictions. Endogenous cell therapy will be HLA restricted by virtue of what I just explained. TIL therapy has been proven effective for melanoma. Endogenous T cell therapy is being studied. And of course, both of these are very time and resource intensive. If you think about a surgery and then a good manufacturing facility to grow up T cells, very, very expensive, time consuming. It takes weeks, if not a couple of months to grow up T cells. And so there can be delays and possibly even toxicity. So with the background that, we know TIL has been used as treatment for metastatic uveal melanoma, this is Dr. Kamula's experience at the NCI, 27 harvests. 6 patients did not get the treatment, but of the 20 who did get treatment and were evaluable, 35% showed tumor reduction by at least 30%. We asked about harvesting from the eye. So prior to 2008, few uveal melanoma patients at MD Anderson were treated with TIL, but when we did treat them with TIL, it was TIL grown from liver mets. Well, what about these eye tumors here? On the left, you see an enucleated specimen with a fairly big tumor. It's not the same one you see here, but you see a tumor inside an eye here. And then you see another uveal melanoma enucleated specimen with a much smaller size tumor, but it's near the front of the eye. It's in the ciliary body portion of the eye. Can we grow TIL from there? I mean, the bottom line is yes, but historically, we had impaired TIL expansion of uveal compared to skin melanoma. And remember, I said we were harvesting mainly from the liver before 2008. And when we were harvesting in the liver in 33 cases, we essentially were only successful about 45% of the time. Compare that with skin melanoma, where it was successful about 2/3 of the time. And that skin melanoma rate has just gotten better and better, frankly, over the years. Also, when we were able to grow from those 15 patients in uveal melanoma, we actually expanded far less number of T cells than we did in skin melanoma. Uveal melanoma, expanding 100, 150,000 T cells on average, 1 million T cells on average, where cutaneous melanomas would often get far more than that, 30 million or so. I meant to say 10 million in uveal and about 30 million in cutaneous. So we actually wrote an amendment to say, could we actually harvest from primary tumors, and what would that look like? And so the short answer is yes, we can harvest from primary. We harvested 3 primary tumors and 24 metastatic tumors in a more contemporary data set and found that, I take that back, we harvested 20 primary tumors and 53 metastatic tumors in a more contemporary data set and found that we were successful in about 15% of primaries and still holding pretty steady at that 45% metastatics. And what we learned from those that we were able to grow from, not much. I mean, they were small, medium tumors, but we don't really think size affects your ability to grow TIL. The GEP class was class 2 in 2 of them, and 1 was unknown and 1 successful TIL harvest. And of course, Caucasian and blue eyes, which is fairly common demographics. And when we were able to successfully grow from metastatic tumors, they were most commonly from skin subcutaneous because of access, really, and then liver, lung, and lymph nodes. And that's really all I wanted to say. Yeah. You know, in conclusion, what we found with TIL is you can grow TIL from primary uveal melanomas, but as you notice, it was, 15% success rate in 20 cases. It really wasn't that great. And so with traditional conventional TIL methods, the metastatic harvests still have a better, higher rate of success. And those successes were really more commonly from extrahepatic sites, so skin soft tissue biopsies. But in the future, we are employing new methods for TIL growth that actually we have already seen has improved the success of ex vivo harvesting, and upfront TIL harvesting from primary uveal may actually be a therapeutic option. So if you were already thinking about getting your eye taken out for treatment, we can grow those T cells and keep them frozen to be ready if and when the cancer returns. The TIL 3.0 method results in a better type of T cells that we've seen grown. And essentially what 3.0 is now we grow those T cells with 3 different signals outside of the body, and instead of the typical kind of TCR receptor tickling, if you will, and then IL-2, the 2 signal method. 3 signals stimulates the T cells better. We get faster cultures. We get faster time in culture, which you see kind of over here on the far right with uveal melanoma. It used to take us 20, maybe 35 days to get cultured, and we would get kind of a low quantity, as I mentioned, 100 times 10 to the 6th number of cells. And I'm just thinking in my mind what that is, 100 million. Did I say 10 million earlier? I think that's 100 million, right? 10 to the 6th is a million times 100. Yeah, so 100 million. Apologies, guys, it's late. And now with the new method where you see in squares, we were actually growing the T cells not only sooner by day 15, but we were getting far greater quantities up in the 200 millions quantity of cells. And then what you see in the bottom is we're getting a better ratio, so better effector T cell to regulatory ratio. So I mean, the short answer is we can grow better with the 3.0 method than we can with the traditional method, and that's evidenced. It's evidence here. So when you see here the TIL 3.0 method, 97% of those cultures expanded. And just to tell you how many that is, that was it's 97% of 32. Again, apologies that I didn't put the math on the slide. But 97% of those 32 successfully grew, and 34% in the kind of traditional 2 signal method. And, what was the number of T cells? Again, in the high 200s, 291 million compared to less than 100 million in traditional. And they grew faster in 18.5 days on average compared to 29 days. That might actually be a median, not an average, which is even better. And then here on the right, what you see is traditionally 88% of the time from primary uveal melanomas, we were unsuccessful, only successful growing it about 12% of the time from primary tumors, about 42% of the time from metastatics. That is reversed essentially with the TIL 3.0 method. 90% of primary tumors out of 10 cases we harvested grew. 9 out of 10 of them grew well. And 100% every single metastatic tumor that we harvested of this 22 grew. So pretty great. So that's actually 31 and 32, right? I don't trust my math right now. It's pretty late. Okay, endogenous T cell therapy. Last comment about endogenous T cell therapy. Remember, this is HLA restricted. We get it from your peripheral blood. We enrich those T cells. We reinfuse. We must know your HLA type, and we must know the peptide that we're after or the antigen that we're after. And there are a couple of options for this trial. for these options. SLC45A2 is a known melanosomal protein in uveal melanoma. It's actually a mutation in this protein, or the absence of this melanosomal transport protein, that's responsible for making the white tiger white, this mutation in one singular gene. And this gene is expressed in uveal melanoma. In fact, we think it's overexpressed. And so we are targeting that with autologous T cells, again, using endogenous T cell therapy. Our trial is restricted to A0201 and A2402. And by the way, you get one HLA allele from your mother and one from your father. So you have two chances historically to be eligible when there are when there's a criteria. So here you have essentially A0201 or A2402 for eligibility. We're targeting SLC45A2. We do have an amendment planned for growing these T cells against PRAME target. We're doing the pulse peptides right now. I do want to comment, you do have to stay in Houston for this study for about 5 weeks, just FYI. And then endogenous cell therapy. This is just another close-up schema of that same HLA trial for SLC45A2. You've got metastasis, and we do HLA typing. If you're O201 or 2402, we then take your white blood cells out of the body. So this is white blood cells coming out of the body, leukapheresis. We go looking for those antigen-specific T cells with the T cell receptor that recognizes that specific protein in the context of this HLA molecule, either SLC protein or PRAME protein. And then in Turnstile 2, we are expanding those T cells, giving you chemotherapy, and then re-infusing those T cells in the body, and then giving you some checkpoint blockade for 4 rounds. You take some interleukin-2 shots as well, twice a day for the first couple of weeks. So again, how to make T cells, either from a tumor or from the blood, and there are ongoing study efforts. TIL therapies going on at University of Pittsburgh Medical Center with Dr. Kamula, and we've got the SLC45A2 and soon to be amended PRAME trial at MD Anderson. Okay, that was a lot. So there are a number of completed trials, as I mentioned, and we're anxious to see the results from those studies. But what I hope you see is that there are several dedicated uveal melanoma trial options that exist even during this pandemic of 2020. And so we have far more options right now during a pandemic than we had even 5 years ago, let alone 10 years ago. And really, autologous T cell therapies, in my opinion, are having their day, as are intratumoral therapies. I think these are two of the most exciting treatments that we have right now. So with that, I will thank you, and I appreciate your attention.